Lisa as well. We'll look forward to hearing from you.
Okay. Thank you all for calling in, listening, having us on this call. So as Jenny said, I'll be presenting the ClinGen RASopathy Expert Panel's Gene Curation project, which started in 2016 and has not concluded, but has come to a short pause after our submission for publication in the ClinGen Special Issue in Human Mutation. So just quickly, I'd like to show the members of our expert panel. Lisa Vincent and Bruce Gelb are the chairs of our group. Daniel Azzaretti is our coordinator, and Brandon Cushman and I were the primary biocurators assembling the evidence for all the gene disease associations. And then we have several other different expert panel members from around the world, clinicians and physicians at different labs and hospitals who helped on their expertise on looking at some of the literature and scoring some of the cases. So the goals of our group to start were, the first goal was the variant curation effort, which we began and published a paper on earlier this year, which just includes specification of the rules, the ACMG, specification of the ACMG guideline codes as they pertain to RASopathy variants. We also have several different expert panel classifications of these variants in ClinVar that went in with that publication with the goal of resolving discrepancies in ClinVar and providing interpretations that sort of are held to a higher standard than some of the other classifications for clinical significance variants. So the project we're talking about here, though, is our Gene Curation project, which included the curation of evidence using the ClinGen Gene Disease Clinical Validity Framework. We curated evidence for 19 different RASopathy gene associations with 5 different distinct RASopathy conditions. The genes are here on the right, and the phenotypes as well. So we curated for Noonan syndrome, cardiovascular syndrome, Cell syndrome, Noonan syndrome with multiple antigenies, as well as Noonan-like syndrome with loose anagen hair for all 19 of these genes. So 95 associations total. Before I get into the actual process or any of our results, I'd just like to give a quick background on the RASopathies themselves. So RASopathies are a complex group of diseases that are associated with alterations in the RAS-MAPK pathway. Alterations in these genes lead to multiple congenital anomalies. For example, different craniofacial anomalies and distinct facial features for each condition, different cardiovascular abnormalities, short stature, neurologic and oncologic manifestations, for example, susceptibility to tumors, different skin and hair anomalies, such as the loose anagen hair, the rough hair of the NFLAH syndrome, intellectual deficits that range anywhere from mild to severe that are not associated with all RASopathies but are somewhat common, and then different nervous and musculoskeletal abnormalities such as seizures, and then some patients also exhibit vision impairment. On the right here we have the map of the pathway provided by one of the reviews, and this sort of highlights some of the, or I guess it highlights the different associations for each gene as of 2013. I don't know how in-depth their curation process was, but it is kind of just a nice map of which gene is traditionally thought to be associated with which condition. This is sort of a very zoomed out overview of our results. This is just the graph of the highest association for each of the 19 genes with any RASopathy. As you can see, most of our genes are definitive genes. RASopathy pathway is a very or the RASopathies are a very well-studied group of disorders, and the genes are very well-studied as well. And so there are a lot of different definitive genes. There are also a few strong genes that we found that met the amount of genetic evidence required to classify them as definitive, but are a little bit newer associations, and so they hadn't quite met the time requirement, the 3 years before the first publication, and one of them only had one publication as well, so it didn't meet the requirement of having 2 publications. But there were a few other genes that were limited or moderate, and actually 2 newer associations that we actually disputed the claims to their association with RASopathies. But I'll get more into the details of those a bit later. So just to go over our workflow, I think we were pretty consistent with a lot of the groups, the gene curation groups that we did have sort of our own process that was developed in terms of the finalization of the curations. That was a little bit different. So after we developed our list of genes for curation, Brandon and I did the primary biocuration for all of the gene disease associations. We then brought the evidence and the classifications to the expert panel calls where we reviewed the evidence and discussed the evidence associated with all of the different conditions and diseases. And then after each call, we would send out a survey surveying the group members whether they agreed or disagreed with our classifications or whether they would provide a different clinical validity or had any comments about the associations themselves. And if we reached a consensus of 80% or more based on the Google survey, then we finalized the clinical validity classification. But if there was not a consensus reached, then we brought it back to the group and discussed whether perhaps the limited evidence that had been published should potentially be disputed, or perhaps this is a newer gene disease association that doesn't quite have enough literature evidence currently published to get to where the field feels it should be, and so maybe we need to look at internal case data and see if we can provide ClinVar submissions in our supplement in order to publish these cases ourselves, so that, or these variants ourselves, so that we can refine the association's clinical validity to where it will be, in the next year or 2, so we don't have to go back and curate it when all those cases get published anyways. So we then bring it back to the group and discuss again, have another survey, and then finalize. And so before I get into the results, I'll go into some of the major challenges, and hopefully this is helpful for any other curators on the call that we had sort of dealt with. The first problem, one of the major problems in the field itself, is that a lot of the literature that we curated evidence for actually contains cases that don't really have a whole lot of phenotypic information provided, or don't necessarily provide the actual phenotypic information required to differentiate, for example, Noonan syndrome from CFC syndrome, or just because some of the cases are really young patients that haven't developed, some of the age-dependent features that are associated with the condition. And so due to this variability and overlap, it brought us to the question as to whether we should lump or split. I will say that the RASopathies are considered and treated by clinical experts in the field to be distinct conditions, so we really felt like we should split the conditions. But I just wanted to introduce this issue before I talk about our other issue, and then I'll talk about how we decided whether we should lump or split. So the other big problem that we had is that a lot of the functional evidence that is provided in the literature actually doesn't necessarily implicate one specific condition over another. A lot of these assays measure the same endpoint and really don't indicate anything different about the molecular mechanism of the disease or whether it's actually a Noonan variant versus a Costello variant. So just some quick examples of this. A lot of the ways that the papers would show the variant impact was to do assays of the variant's impact on changes in phosphorylation of different proteins in the pathway. Most of the genes that we studied, alterations to them caused gain of function of the pathway, which is pathogenic. And so just measuring, you know, ERK is, it has increased phosphorylation due to this variant. That could happen from, you know, a Noonan variant in KRAS or a CFC variant in KRAS, and there's really no differentiation between the two. The other issue is that the animal models, given that there's enough overlap phenotypically in humans, having animal models that display, you know, some of the major features of RASopathies like short stature, cardiovascular anomalies, or the craniofacial anomalies really don't provide strong evidence either way, even if it is a variant that has only been associated with one specific condition. So we felt that it was a little bit troublesome to actually use that as evidence to score, to actually have a separate implication if we were going to split, which we did end up splitting. Then the other thing is that a lot of the biochemical function and protein interaction assays that are done in other disease domains aren't super relevant for the focus of our project, because all of these genes are known to, or almost all of these genes are known to be associated with the pathway, and it's really the intricacies of how they interact with the pathway that end up causing the different phenotypes. And so just having that it's implicated in the pathway or it interacts with this protein is kind of like, well, we already know that. And so we decided to be stringent about the biochemical function and the protein interaction assays as well. I will say there are some exceptions to the lack of specificity of some of these assays. For example, P2PN11 NSML variants have been shown to have hypomorphic alleles. They're shown to be hypomorphic alleles, and that they actually decrease phosphorylation of certain proteins, which is, I think this was the only example we had of that for the genes that we curated, but almost all of the genes that we curated have gain-of-function mechanisms of pathogenicity. And then there was also an HRAS mouse model that had features that are more specific to Costello that would be an example of something that we felt was scoreable. So back to the question of whether we should lump or split, because we dealt with this for a long time and the field itself is still dealing with it. But we felt that we shouldn't just replace all of the terms with a RASopathy curate, because we felt that we would lose the actual utility of our work. So we sort of took a long, hard look at the lumping and splitting assessments for each of these and ended up deciding to split. But just to give you an example of sort of why this was so troublesome for us. So BRAF has been associated with cardiovascular cutaneous syndrome as well as Noonan syndrome. They are clinically distinct conditions that are treated differently, have a different outlook, but the inheritance pattern is the same. The molecular mechanism is either not different, like truly not different, or has not been discovered to be unique yet. And assessing whether the phenotypes are actually variable enough to call them separate disorders is also questionable given the overlap. But since they are, presented to patients and their families as different conditions, and they are treated differently, we felt that this outweighed the fact that we don't yet know what makes these conditions quite different for this, and we felt that it was important to maintain the current nosology in the field. The other example that actually supports our ability to split is P2PN11 and Noonan syndrome versus NSML. As I had said, a few of the variants that cause NSML actually have a different mechanism of disease in that they are hypomorphic alleles. And so given that there is that distinction, we would be able to split via lumping and splitting guidelines verbatim. And so we felt like losing the granularity for that would not have been helpful either. And there actually P2PN11 is definitive for both Noonan and NSML. And so our overall conclusion, whether to lump or split, was to split. As I said, with the rationale that even though someday the RASopathies may end up deciding to lump some conditions into one entity, our current belief is that they will discover eventually that there are some modifications or different mechanisms that are underlying these differences, and so we felt it was important to curate in the context of the current nosology of the disease domain and split. So in doing that, we created the small issue that, not really an issue, but we ended up having to look at a lot of different curations that were sort of around the limited, no evidence, disputed, refuted categories, which I know a lot of groups are struggling with right now because while we have the definitions here, when you get into the actual examples, it's hard to know where to categorize everything. So I'll just go through really briefly how we define them, and then I'll give an example of how we defined what was disputed versus no evidence or limited. So typically limited is just thought of not having any contradictory evidence, but there is scorable evidence, not enough to get to moderate, but it is not necessarily disputable. No evidence being there's actually no pathogenic variants that have been identified in humans. There's no convincing, or not even, I shouldn't even say convincing, there's no evidence or claims that a variant identified in the gene has caused disease. And then disputed being that there has been a claim that a variant is causing a disease or may be causing a disease, but some evidence is somehow disputing this role. And then refuted being the same thing, except that all of the evidence that has been presented has been refuted, and that there is no evidence supporting the association remaining. And we didn't actually use refuted because we sort of thought of refuted as that there actually had to be a published paper or study showing that these variants do not cause this, which we don't have in the RASopathies. But we did have several that were disputed. And so, as I had said, a lot of part of this splitting out was having these disputed claims. And one example of this is that a lot of genes had certain patients that had been described as Costello-like patients, but they were really young, and they didn't really have any actual differentiating features to show that they actually were Costello versus, Noonan or CFC. And also the field feels very strongly that Costello syndrome should only be associated with HRAS. So we disputed these claims, all of these claims, because actually there might have been one we didn't. But I think it was NRAS. We didn't dispute its association with Costello, but a lot of the Costello claims didn't have either a strong claim in the sense that the authors didn't feel as though they actually had a definitive diagnosis for the patient at that time, and they would say that, and we decided to dispute that, or the evidence they provided wasn't convincing enough to count the patient for that disorder, or the variant itself just didn't have enough evidence to support its pathogenicity, indicating that it may be some other variant in a different gene causing these Costello-like features or other features. And so I will note, if there was no claim present, we just called it no evidence. The claim had to be there in order to call it disputed. So an example of this is the SOS1 and cardiofasciocutaneous syndrome curation. This first patient from Narumi 2008 had a 3 amino acid insertion between proline 481 and glycine 482 in SOS1 gene. And they actually very well could be a CFC patient. They're 8 years old, and they were recruited from a CFC cohort, but there was no further evidence for this variant's pathogenicity, and only 6 genes were sequenced. So we didn't feel like this was convincing enough to score. The other next patient had a diagnosis that was described as Noonan syndrome or CFC syndrome, and we decided to dispute this since it was a weaker claim. The authors weren't providing any detailed evidence indicating that this was a CFC patient. And then there was one more from this paper that had a variant that actually was functionally characterized. There would be enough evidence normally to score it as a half a point based on variant pathogenicity, but the patient was atypical for Noonan and CFC, according to them, even though they did resemble intellectual disability that is more often associated with CFC. So we didn't score that case. This next case had another variant that was functionally characterized. It had been seen, I think, in other patients as well. No, I think it was just functionally characterized. But the case, the individual did not actually have intellectual disability present, and there weren't really a whole lot of other features presented in the paper to indicate that this actually was a CFC patient. So we didn't trust that this case was enough evidence to support the association. So we didn't score that case either. And then this last variant also didn't have variant evidence, and the diagnosis was described as inconclusive between CFC and CS. So I just thought that was a good example of sort of all the different ways that we looked at variants and decided whether they were scorable cases or not. And I will note this. Sorry, did someone have a question?
I did, Andrew. It's Courtney. I just had a question on the first one. You had said they were only tested for 6 other genes. How often in these RASopathy genes do you see oligogenic variation within the RAS pathway?
Sorry, what was it? Variation?
Meaning, how often has it been reported in literature that an individual with a RASopathy has variation or pathogenic variants in more than one of the genes associated with RASopathy?
Very little, if at all.
Okay. Yeah, because I was just trying to see if it's already little, would that be something where you wouldn't want to count a case like this because if it's not something you're used to seeing, just trying to evaluate that kind of testing if you saw it a lot or not. Thank you.
Okay. Yeah. The other thing about this curation was that there were, as I had said, like some of these variants were functionally characterized, so there were potentially functional studies that could have been scored if we had been scoring the variant specific functional studies, but we felt that this wasn't, since there was no evidence in humans that was convincing, that we would dispute this claim that SOS1 causes CFC. On the other hand, we have an example of a weak association that we classified as limited, which was NRAS and CFC. So there was actually only one publication supporting this association. The first patient had features consistent with CFC, but they also had a duplication, a large duplication that was modifying the phenotype. They presented with hypoxic ischemic encephalopathy, and so we couldn't score that case due to the modification. And then there was this other case that had the threonine 50 isoleucine variant, which has actually been seen in patients with Noonan syndrome that also had mild developmental problems and had been functionally characterized. And given that this patient was characterized as a CFC patient, they're 11 years old, they do have intellectual disability, we felt that there really wasn't enough reason to actually dispute this particular case, and so we felt that this association would be limited. So we then had the caveat in our, given that we think that this case may very well, this variant may very well be causing the same disorder across these Noonan syndrome patients that also have mild developmental problems, we felt that it was important to highlight that over time this association may end up being disputed if the mechanisms underlying this variant turn out to be the same. And so that is our lumping and splitting issues. The second issue was our experimental evidence scoring issues. And so, as I said before, a lot of the experimental evidence actually wasn't specific to a clinical phenotype. An example of this is the KRAS Noonan mouse model. So this variant, the PV14I variant, was a knock-in in a mouse model. It had been seen in a patient with Noonan syndrome, and the mouse developed features that were consistent with RASopathy in the sense that they had, the mice had short stature. This is a graph of their growth over time, and they had reduced survival rates over time. They also had craniofacial anomalies shown by the X-ray. They also had cardiac hyperplasia, valvular hypertrophy, splenomegaly, and myoproliferative disorders. So if this was a model that was being scored for RASopathy, this would be a really good mouse model, and we would probably classify it with a default, or score it with a default 2 points. But given that none of these features really differentiated between Noonan and the other features, even though the variant had been seen in only Noonan patients, we were a little bit troubled with the logic of applying the evidence. And I'll talk about what we ended up doing with all these, but we actually ended up scoring this as a half a point instead of 2 for Noonan only. So the other example is the functional alteration studies that I had talked about. So this is an example of, the data they provide showing gain of function in the pathway. Again, these are variants that have been seen in patients with only this variant, or maybe even the variant has been seen across both conditions. And so we can't really say that that variant doesn't also cause the phenotypes that we're seeing that are associated with that gene, or both phenotypes, I should say. So we felt that these studies weren't really in support of any particular phenotype, given that they were measuring gain of function in the pathway. It's more implicates the gene's association with the RASopathy pathway than it does with a particular condition. So we largely did not score these. The one exception was the NSML example that I had mentioned. So before I sort of show what we ended up doing with all of the different functional evidence, I just want to go through what we originally had thought of and why we decided not to do that. We had originally had the idea of providing a score for the gene's association with RASopathy that included all the experimental evidence, and then providing the score for each specific condition association, so like NRAS and Noonan, NRAS and CFC, NRAS and NSML, that didn't include the experimental data because it largely didn't implicate those phenotypes alone or conditions alone. And so that would look like having BRAF with RASopathy, definitive 18 points, lots of different cases, lots of de novo cases, or I should say lots of different variants. Lots of models, zebrafish models and mouse models have been found. And then you would have BRAF for CFC would still be definitive, but only based on the genetic evidence, and BRAF for Noonan syndrome would be moderate, but only based on the genetic evidence. But we discussed this a lot on the gene curation call, and we chose to actually just downgrade the functional evidence instead, so that we would be following more closely in line with the ClinGen gene curation framework instead of providing, blanket restrictions on our different associations. So what we ended up doing was, as I said, we downgraded a lot of things. So the first evidence type is biochemical function, protein interaction, as I mentioned. We minimized, or not minimized, but we limited the amount of evidence that each gene could attain for this, because all of these assays are basically just implicating the fact that this is a RASopathy gene, and it's not really differentiating between any condition. So all genes except for LZTR1 ended up getting a half a point based on these well-established functions showing that, or experiments showing that they are RASopathy genes. We ended up not scoring any expression points. I don't think there even were any studies that were trying to show that RASopathy genes expressions was specific to a condition, because the pathway is ubiquitously expressed in organisms, and so any tissue-specific expression wasn't relevant for us. I've mentioned a few times the functional alteration studies typically didn't score any points because the variant showing a gain of function in the pathway is a common mechanism across all of the different conditions, and it really shouldn't count for any one in particular. As for the mouse models or animal models, we typically downgraded what would have been, a default model for, say, the general RASopathy phenotype to a half a point, just because they were variants that were seen, only in patients with those that condition. So we felt that would help us decide where to put the 5.5 points, but we felt like it should be downgraded given given the situation. So some quick examples, and then I'll get to the results of our project as a whole. So this functional alteration study was a variant that had been seen in a CFC patient, and instead of using this as, experimental evidence, we just used pretty much all of these studies as evidence for variant pathogenicity and didn't double count. And that's pretty common across a lot of groups, but we always put it on the variant. And so this other mouse model, or sorry, not a mouse model, this is a worm model. So we normally might have given this worm the downgrade of 1 point if it was, for a RASopathy, but since it was the variant that's associated with SHOC2 and NSLAH, we gave that curation 0.25 points for this. And then this is the example of the PTPN11 NSNL variants that were shown to actually abolish phosphatase activity instead of causing gain of function. And so we gave that the full ½ point for heterologous expression of variants in non-patient cells. And then the BRAF-CFC mouse model is an example of really common variant found in BRAF patients being downgraded to 0.5 points because it's not necessarily showing features that are only consistent with KRAS. So, results of our project. So I'd already shown this figure, this one on the left showing all of our highest RASopathy association with each of the 19 genes. This one on the right showing all of the curations and their associations. It's all 95. As you can see, a lot of them are no evidence. And that was because a lot of the genes themselves have only been implicated with, 1 or 2 or maybe even 3 conditions. And so we did the literature search for any evidence or any claims for all of the associations because we felt it was, important to know. But most of them ended up being no evidence. As I mentioned, we had a lot of disputed claims. We had a decent amount of limited as well. And you'll notice that there's actually 2 more definitives, 2 genes that have multiple definitive associations. One of them is PTPN11 with Noonan syndrome and NSNL, and I'm forgetting the other one right now, but I will show the table in a minute. So the other thing I wanted to show was all the conditions that we curated for and the number of genes associated with each condition. So most of our genes are associated with just Noonan syndrome. And the one on the right, or the graph on the left, I should say, is just showing all of the genes that had strong or definitive associations. The next most common was CFC. There's only 1 gene that's definitively associated with Costello and NSNL, and there are 2 with Noonan syndrome and loose engine hair. Then this graph on the right is anything limited and up, so anything that has a claim that we didn't dispute. Again, Noonan syndrome was the most common gene, but a few more genes have been implicated with NSML, and then NRAS actually had 1 or 2 cases, I can't remember, that we couldn't dispute for Costello syndrome, so that was why Costello syndrome had 1 more association. So this is all of our final data. It is still pending review and revision, so it is not published yet. But these are our final conclusions for each gene. And we wanted to present this more on like a gene level and show how each gene is associated, or which conditions each gene is associated with. And as I had mentioned before, when we were considering having a RASopathy association, we had originally had a column that was the RASopathy curation, and it had just its own score. But given the current display on the website, this would potentially be its own page, and it might be troublesome if BRAF is considered definitive for RASopathies, and it kind of doesn't show that it's disputed for Costello and limited for NSML. So we hope that at some point the website goes to a more gene-based approach, the ClinGen website, and this will sort of be able to be outlined a little bit more clearly, but we felt this was the best way, given we had so many curations to display it. We also have in our supplement summary evidence blurbs describing all of the different things that we found for each gene disease association, all of the different caveats we wanted to present. So for example, NRAS is one of the genes that's a little bit newer associated. We couldn't get any internal data to be published, but some of the labs in our expert panel members are members of do have unpublished cases that will eventually be, seen in NRAS. And so it's very much like on its way up for its RASopathy and Noonan association, and so we felt like it was important to have that somewhere in our paper, and so we put it in these summary evidence blurbs. And likewise, there's some genes that are going down. I think RASA2 is one of them. But yeah, so that was all of our results. Just to bring it back to broader conclusions before you have any questions. So a total of 95 curations were performed, 19 genes on 5 distinct conditions. Our goal is to have this level of granularity to hopefully inform clinicians. This is the amount, this is what the evidence that's out there for BRAF association with Noonan, even though it's a CFC gene. This is how we scored this evidence, and you should be aware of how we thought of the current strength of it. And we hope that it's helpful in if a variant that has been associated with a condition that's not the traditional association is found, like what are these patients, like what are the conditions that should be noted that have been implicated with the gene, not necessarily that this patient's at risk for, but what's out there. As I said, our work is currently undergoing review and hopefully going to be published in the Human Mutation Special issue. And one of our biggest takeaways from our process is that hopefully ClinGen will discuss further specifications on how experimental evidence should be scored for genes that have multiple associations. While we feel like this didn't change any of our classifications, whether you apply with functional evidence or not, it may in other disease associations or disease domains, and so we feel like it's important for curators to have pretty strict guidance on where they can allocate this evidence. And then last but not least, we do have some more genes. I think we have like 5 or 6 more that are on the dock for curation that we will hopefully get to in the next few months. But other than that, I would just like to thank the RASopathy expert panel, the Gene Curation Working Group, the Clinical Domain Oversight Committee, and the Limping and Splinting Working Group for all of their guidance in developing our curation process and going through our edits. I'd also like a special thanks to Brandon Cushman, Lisa Vincent, Martin Zenker, Marina DeStefano, Heidi Ream, Jonathan Berg, and Courtney Saxon for their efforts and their feedback through the finalization of our process. And with that, if anyone has any questions or wants me to go back to any slides, just let me know.
Hi, great presentation. This is Ana Morales, and I am curious. I do cardiovascular genetics. In your literature search, did you come across any evidence for any of these RASopathy genes associated with isolated cardiomyopathy or a cardiomyopathy dominant phenotype where the other syndromic features may be mild or completely absent?
So yes, I believe it was RASA1 that had been associated with different cardiac anomalies and cardiovascular anomalies, and it had been suggested that it may be a RASopathy phenotype in the literature, but there weren't really any features that couldn't be explained by, you know, a second variant in the patient that had syndromic features. So that was one of the ones that we think is just an isolated cardiovascular gene.
And that's RASA1?
Yeah.
Thank you.
We actually have been working with the HCM group, and we've been sharing our gene curations with them. And it is really felt by the expert community that none of these genes are currently associated with isolated HCM.
That's great. Thank you.
Well, thanks so much, Andy. That was a really great presentation and definitely shows the complexities of trying to decide on the lumping and splitting and where to count the experimental evidence. I guess I had a question about if you had decided not to include, for example, a functional alteration experiment in the experimental section, did you then include that as variant or case level evidence if you felt like it wasn't specific? Okay.
Yeah, so we typically, we always use that as evidence for the variant pathogenicity, and if there was enough, you know, phenotypic evidence that the patient could still be put into that curation, then we scored it. But yeah, we felt that it was more for the pathogenicity of the variant and not for the experimental
disease. Yeah. And I appreciate you bringing up that, you know, how hard it can be to decide whether experimental evidence is specific to a disease phenotype and needing more guidance on that. And that's certainly something that we can work on maybe in the Gene Curation Working Group. I know this just kind of come up before, but, you know, certain pieces of evidence that might be fine to include in more than one curation, like let's say it's for a heart condition and you know, well, there are a couple of heart conditions that a gene has been associated with. It may be fine to include expression data for both of those, but I can see for, yeah, if you have a specific variant that's being expressed in a mouse model and you're not really sure, you know, the mouse is showing certain features, but you don't really know if it's specific to one disease or another, that's kind of challenging to know where it should be counted. So we can definitely talk more about that. Does anyone else in the group come up